Today we're going to be discussing mutations. There's a video here linked. Please make sure you watch that before watching this video. So mutations, we know, are any inheritable change in a sequence of DNA. These changes might be a substitution of a nucleotide, a small deletion or insertion, or it could be a larger alteration affecting a whole segment of a chromosome. The mutation must persist through DNA replication and be transmitted during cell division, so it must be passed down from generation to generation. These mutations in DNA can then be transmitted to mRNA and then to proteins, where they could affect the form and function of an organism or even its phenotype. Point mutations are the simplest type of mutation because it's a change in just a single nucleotide. For example, replacing thymine with another base like cytosine. These types of mutations are very common, but many point mutations have no detectable effects on an organism because the mutation likely occurs in regions of the genome that doesn't even encode for a protein. Point mutations that do occur in coding sequences of genes do have consequences for an organism because these are transcribed to mRNA and translated to a protein, and just one nucleotide being changed, one base, can alter an amino acid sequence and therefore a protein. On this slide, we can see a DNA sequence coding for beta globin, which is a subunit of hemoglobin that carries oxygen in red blood cells. So here we could see a non-mutant form. So we would see that the translation would be a proline, a glutamine, and a glutamine. A silent mutation occurs when we could see A with T, and we could see that changed to an A. So a GAA would still code for a glutamine. So even though there was a mutation here, it still coded for the same amino acid. So we would call that a silent mutation because the mutation happened, but it didn't cause an effect on our amino acid sequence. A missense or non-synonymous mutation would be here where T and we have an A. So the T would pair with a U instead of an A. So it should be an A here, but it is a U. And GUG codes for a valine instead of a glutamine, as we could see here in the non-mutant form. So proline, valine, glutamine, that would change the amino acid sequence and therefore change the protein and therefore the ability of hemoglobin to function normally. So that's called a missense mutation. A nonsense mutation is a mutation that can occur that changes what should be an amino acid to a stop codon, which will terminate translation and produces a shortened protein, which typically always have some sort of harmful effects. Small insertion and deletion mutations can be very common types, which deal with small numbers of nucleotides. In non-coding DNA, these mutations have little to no effect, but in protein coding regions, these can cause a large effect depending on their size. Cystic fibrosis is actually an example of a mutation caused by a deletion of only 3 nucleotides. Small deletions or insertions that are not in exact multiples of 3 can cause major changes in an amino acid sequence because it causes what's called a frame shift mutation. So as we can see, the first, we're going to look at it in terms of a sentence. The sentence, "The big dog saw the cat eat the bug." If we got rid of the E there, we have that small deletion of the E. Now, our sentence no longer makes sense. Everything was kind of shifted over to the left. When something is deleted, it causes a new reading frame or a frame shift. And an insertion would cause a similar effect because it would shift everything over to the right. This slide shows the consequences of a frame shift mutation in that beta globin gene we were talking about before. So here's the non-mutant form and the amino acid sequence there. Here we have an insertion of a cytosine guanine base pair, and therefore it shifts the entire frame shift to the right, and therefore our amino acid sequence we can now see is completely off besides that very first amino acid. So that causes an entire different protein that could be created. An important source of new mutations in many organisms is the insertion of movable DNA sequences into or near a gene. These movable sequences are called transposable elements or transposons, and they're actually able to jump around the genome, leaving one place and inserting into another. The genomes of virtually all organisms contain several types of transposable elements, each present in multiple copies per genome. Chromosomal mutations are mutations that affect larger regions, extending over hundreds of thousands or even millions of nucleotides, and have effects on chromosome structure that are often large enough to even be visible through an ordinary light microscope. When a pair of chromosomes fail to separate during anaphase of cell division, it's known as non-disjunction. The result is that one daughter cell receives an extra copy of chromosome, and the other daughter cell receives no copy of that chromosome. It's very common in humans, but is infrequently observed because the results are typically lethal, meaning pregnancy would end in miscarriage. There are a few major chromosomal differences that are found in the general population, and the most familiar is Down syndrome. There are two other trisomies of autosomes that are sometimes found in live births, trisomy 13 and trisomy 18. Both are much more rare than Down syndrome, and the effects are more severe. In both cases, the developmental abnormalities are so profound that newborns with these conditions usually do not survive the first year of life. An abnormal number of autosomes is actually pretty rare, but an extra or missing sex chromosome is much more common in live births. Examples being Klinefelter syndrome and Turner syndrome, which we talked about, especially back in biology class. There are more extreme mutations that result in changes to the number of complete sets of chromosomes called ploidy, which refers to the number of sets of chromosomes. Humans are diploid because they have two sets of 23 chromosomes, or 46 in total. Polyploidy describes more than two sets of chromosomes, and that's typically lethal in humans and other animals, but is very common in plants. We can describe mutations in two different ways, depending on genotypes and phenotypes. Change at the level of DNA, we would obviously call it a genotype, and the effect of the mutation on the traits of a cell or an organism would be the phenotype. However, we know that the relationship between genotype and phenotype is not always straightforward, because when mutations occur in non-coding regions of the genome, they typically don't have any observable effect on a phenotype. Silent mutations in coding regions do this too. Some mutations in genes can have dramatic effects on an organism: missense mutations, nonsense mutations, or frameshift mutations, and even extra or missing chromosomes. The change in the genotype does lead, then, to a change in the phenotype. In diploid organisms, the relationship between genotype and phenotype also depends on whether the mutation is homozygous or heterozygous. So if our dominant allele is non-mutant, and the recessive allele is actually a mutant gene, then someone who is heterozygous for this trait would have the non-mutant phenotype, even though there is a mutant gene present. So over here, homozygous recessive would be the mutant phenotype, so we have two mutant genes, two mutant alleles, and it shows in the phenotype. And here would be the completely non-mutant phenotype and no mutant gene either. Whether a mutation is harmful, neutral, or beneficial, it often depends on factors other than the mutation itself, because remember, it can also depend on the environment. A mutation that is harmful in one environment might not be harmful in another environment. It could be beneficial or even neutral. An example here of this would be our mutation in the beta globin. So say we have alleles A and S. The allele A codes for a glutamic acid in the amino acid sequence, and the S codes for a valine, as we could see over here. Glutamic acid, and then valine. Okay, so only one amino acid would be affected, but this change has a dramatic effect. If someone is SS, they have sickle cell anemia, but if they are AS, they have what's called sickle cell trait. So in Africa, where malaria is widespread, being a heterozygous is actually beneficial because it provides some protection against malaria. A homozygous individual would not have anemia, but also doesn't have protection against malaria. So in this example, S is harmful as homozygous genotype, but it's beneficial as heterozygous in this particular environment. Genetic differences that we see among individuals are ultimately the result of mutations. That's what causes genetic variation. Some mutations result from mistakes that occur during DNA replication, and they're called spontaneous mutations. Other mutations result from damage that occurs to DNA. And that's usually something that you're exposed to in your environment, which we call mutagens. The presence of a mutagen can increase the probability of a mutation by a factor of 100 or even more. Cells have mechanisms to repair DNA damage, but damage that goes unrepaired results in mutations called induced mutations. So they could be spontaneous or they can be induced. Another source of mutations are those transposable elements that we discussed previously. Most mutations that exist have occurred in the past and have been passed along from one generation to the next, but a much smaller number are new, occurring in the lifetime of an individual, whether they're spontaneous or induced. Although mutations are the ultimate source of all genetic variation, recombination by crossing over, followed by segregation of homologous chromosomes during meiotic cell division, shuffles existing mutations to create new combinations. Therefore, recombination and mutations both create new alleles and are therefore considered sources of genetic variation.